Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. In today's episode, we're going to be talking about the first two levels of protein structure, primary and secondary. These early stages of folding are essential for how a protein ultimately takes shape and functions. Starting with the primary structure, which is the amino acid sequence. This is the linear sequence of amino acids in a polypeptide chain. It's determined directly by DNA through transcription and translation. Peptide bonds connect the amino acids, stabilizing the primary structure, and the sequence is read from the N-terminus to C-terminus. Even a single change, like in sickle cell disease, can drastically alter protein function. The primary structure of a protein can be determined by sequencing. This is most easily done using the DNA that coded for that protein. Moving on to secondary structure. Secondary structure is the local structure of neighboring amino acids. It's the result of hydrogen bonding between nearby amino acids, and the two most common secondary structures are alpha helices and beta pleated sheets. Alpha helices, or an alpha helix, is a rod-like structure in which the peptide chain coils clockwise around a central axis. The helix is stabilized by intramolecular hydrogen bonds between a carbonyl oxygen atom and an amide hydrogen atom, 4 residues down in the chain. The side chains of the amino acids point away from the helix core. Moving on to beta pleated sheets. Beta sheets can be parallel or antiparallel, and the peptide chains lie alongside one another, forming rows or strands that are held together by intramolecular hydrogen bonds between carbonyl oxygen atoms on one chain and amide hydrogen atoms on the other chain. Beta sheets assume a pleated or rippled shape to accommodate as many hydrogen bonds as possible, and the side groups of amino acids point above and below the plane. Parallel beta sheets have strands that run in the same direction, so N-terminus to C-terminus, while antiparallel sheets have strand running in opposite directions, alternating between N-terminus to C-terminus and C-terminus to N-terminus. This difference in directionality impacts the hydrogen bonding patterns and overall stability of the structure. Finally, we have some special notes here. Proline introduces kinks in the chain due to its rigid structure, and this is why it's rarely found in the middle of an alpha helix and in the middle of a beta sheet. It can, however, be sometimes found as the first amino residue at the start of an alpha helix, as here it does not disrupt the helix's structure. On beta sheets, proline is normally found on the sharp, tight bends, as its rigidity limits rotational freedom, making it more favorable for the tight, sharp bends in beta sheets. On the other hand, a glycine only has one hydrogen atom as a side chain, making it very small and flexible. This often disrupts regular hydrogen bonding patterns of alpha helices and beta sheets. Glycine is also frequently found in beta turns, but also in loop regions. Glycine is considered a helix breaker in soluble proteins, but it's crucial for helix packing in transmembrane helices. That's it for today's episode. In the next episode, we'll talk about tertiary and quaternary structure.